Tonight, uh, Dr. Dennis Desjardins has generously offered at the very last minute, and by last minute, it's like yesterday at noon, our scheduled speaker took ill and had to cancel. And I can't tell you how much we appreciate you doing this for us. So thank you. And his good friend and colleague from San Francisco State University, J.R. Blair, will introduce him.
Thanks, Mike. So it's my great honor to introduce my friend and mentor, Dr. Dennis Desjardins. Let me give you a little bit of his history, and then we'll talk about our history just a little bit too. Dennis grew up in a mushroom hunting family up in the Northern California near Crescent City, and basically spent weekends with his family hunting the spruce forests and pine forests up there as a child. After a few starts and stops in things like the construction industry and the music business, he's really an excellent saxophonist, by the way, if you ever get a chance to see him play. Then he entered into a program at San Francisco State, got his master's degree there. Hang on, I need my notes. Got his master's degree there in 1983, and his bachelor's degree there got in 1983, and has got a master's degree in 1985, working with another icon of the North American mycology, Dr. Harry Thiers. And then he went off to get his PhD with Dr. Ron Peterson at the University of Tennessee and obtained that in 1989. After a year teaching at Oberlin College, he returned to San Francisco State, which is unusual for people who get their master's degree to come back and become faculty there. But he started in 1990 as a professor there in biology. And so that's where I came in. I came in 1995. I talked to Dennis about getting into his lab, studying mushrooms, and was accepted. And I'm grateful for that. Thanks a lot, Dennis. So I did my thesis on fungi associated with Arctostaphylos in Northern California. Dennis was, by the way, my mycological stepfather. So I actually studied with Dr. Mike Bugh at the Evergreen State College in Washington, and that's what piqued my interest. But I was really grateful to get into Dennis's lab back then. As a mycologist, he has quite a resume, quite a CV, as you can imagine. So he's has named over 300 species of fungi. He's has over 160 publications in journals and scientific journals in mycology, and is a co-author of two popular books, Mushrooms of Hawaii with Dr. Hemis, and California Mushrooms with Mike Wood and Fred Stevens. And he's also, during that time, was awarded the Distinguished Mycologist, the greatest honor of North American mycologists that can be obtained. He's currently he finally had enough of teaching back in 2022, and after starting there in 1990, so over 30 years at San Francisco State. And he's currently a consultant with Sempera Organics, which is a company that uses fungi, mycelium, and fruiting bodies for health, food supplements, probiotics, and all kinds of groovy stuff like that. He's also a champion table tennis player. Oh, wait, sorry, that's a different guy. Sorry. Yeah, he doesn't play table tennis. Anyway, it's my great pleasure to introduce you to Dr. Dennis Desjardins.
I'm not Dennis.
Well, thanks for having me. Definitely, it was last minute. I was expecting to be here to introduce Dr. Brian Perry. But I'm really honored to be the designated hitter for Brian, who's on injured reserve. And I'll do what I can to entertain you tonight. So I had to go back into my computer after saying yes. I should have said no. After saying yes to see if there was something I could actually talk to you about. So I did find something that I thought I would talk to you about. And do you want me to stand here so they can see me? Or if you can. I'd rather stand up and talk than sit down. You see me in the little box? I don't know. I don't see myself here.
Yes, they can see you.
Okay, great. So what I thought I'd do is talk to you a little bit about what it takes to actually document a mushroom diversity in the tropics. Now, Brian was going to talk to you about what's being done now, particularly in North America with the Fundus project to document mushroom diversity, certainly in California, but all through North America. And with a number of non-academic mycologists, all of you, and a few academic mycologists as well, to try and collect the specimens and sequence them all and to add to our growing knowledge of molecular biology of mushrooms, particularly for North America. So this is going to tie into that a little bit. And you'll see that Brian's name is down here as well. So Brian and I have been working together since he was in my lab. He, like JR, he came through as an undergraduate and got his master's with me, then went up and got his PhD at Harvard, and then came back and did a postdoc in my lab for about 4 or 5 years, and then went on and got a job at University of Hawaii. Now he's over at CSU East Bay. So we've been publishing together. We've probably published 35 papers together. And one of the last major projects we did was the project that I'm going to talk to you about. So the little circles you see up there are areas in the world where I've spent considerable time documenting diversity of mushrooms. I have to say I'm one of the luckiest people on the planet because I actually get paid to crawl around in underexplored jungles and search for mushrooms. I mean, that's pretty amazing. I never thought I would do that when I was young, but that's pretty amazing. Oops, let me turn this on. And what I'm going to do is talk to you a little bit about how we do this, how we actually study and document these mushrooms from the tropics. And I'll do that by using a case study. And that's a case study of our project in the West African nation of Sao Tome and Principe. Now, I spoke to this club, I think it was about 8 years ago, on some of the results from this project. And we did this project between 2006 and 2008. So what I want to do is kind of start off a little bit and talk to you, give you a little background about what Sao Tome and Principe is all about, and then talk to you about how it is that we actually go out and collect, identify what a species is, how you determine what a species is, and all that kind of stuff. And then I'll lead you into some of the results that we obtained from this project in Sao Tome and Principe. So let's, okay, so want to do that.
There you go. You may have to do it on this.
What did you undo here? You're not allowing me to advance.
And now I did it. So these work.
Let me try that.
It should point at the computer. You want to point it here.
Okay, it doesn't want to do it.
You point it here. This is your reader.
No, it's going backwards and forwards in one of them. Oh.
Okay. Oh, it's forwards and backwards. There you go.
Technology, isn't it wonderful? Maybe you could just replace me with an AI robot or something. All right, here's, we talk about the country of Sao Tome and Principe as being at the center of the world. So actually, it is just slightly east of zero longitude, and it's almost exactly at zero latitude. So 0, 0, right in the center of the planet. Sao Tome is the larger of the two islands. It's still pretty small. As you can see, about 854 square kilometers. It's only about 13 million years old. They're both volcanic in origin. Principe is the oldest one. It has subsided quite a bit. It's only about 31 million years old. And let's see more about it. The islands were first found basically by the Portuguese in the about 1450s or so. They have dominated the island for the last 500 years, and it wasn't until about 1975 that the island became independent. The Portuguese found the island and used it as a colony for their slave trade. So they brought their slaves from Angola, Guinea-Bissau, Lagos, and a few other areas around there to the island and then shipped them from there, unfortunately, off to America. The language that is spoken there is Portuguese. As you can see, it's relative. Sao Tome is the largest with only about 150,000 people. Principe is a very tiny island, about 6,000 people. Most of everyone there is African American, about 95%. And I'll mention it again, but almost half the population is under 16 years old. So here's just a couple of quick looks at the island. So the highest peak there, it's only about 900 meters, is shown there. And there's a number of these basaltic spires that come out on the island as well. Principe, there's the highest peak there, the Pico de Principe. And again, you see some of the basaltic spires there. This is what the coasts look like. You see the basalt along there as well. If you try and figure out where people are living, they live in very, very small little buildings, little shacks. There are a number of old colonial mansions or large chalets that are on the island that are basically abandoned. Very few tourist resorts. Not many people go there. Here's some more, another abandoned house. And here is some of the population, absolutely beautiful children. This was 2 families worth. They lived near the area. These kids followed us all around the island when we were collecting. That was really a lot of fun. My favorite is this. I love the attitude on your face. The entire government was pretty much run by the sugarcane industry for a long period of time. And then the sugarcane took over in Brazil and Southeast Asia, and that kind of defeated the purpose of growing sugarcane on the island. So they switched to chocolate. And if you've never seen chocolate, it's absolutely beautiful plant, and coffee. And that's what has been running most of the agricultural funds until they discovered oil surrounding the islands in their territorial waters, which is probably going to shift what happens on the islands if they can contain control of it. And that's what the big fight is now, because all the other countries around there in Africa want that oil as well. But hopefully the oil will go to the Sao Tome people and they'll be able to use it. So this project that I'm going to tell you about, the goal of the project was to go to these 2 islands and collect and document all the fleshy fungi that occur on the islands. Now, for me, fleshy fungi means it belongs to the order Agaricales. They're the fleshy mushrooms. Anybody that has studied with me knows I hate polypores. So we didn't look at polypores. Sorry. We didn't look at lichens either, Nikki, so sorry about that. Or bryophytes or any of the other stuff. But we did look at fleshy fungi. So pretty much anything that you would think of as a mushroom, except the polypores, is what we were collecting and documenting, okay? And to just give you an idea, so there, again, the sizes of Sao Tome and Principe, both islands are only about 200 to 250 kilometers from the mainland of Africa, but they have never been connected to the mainland. So they're volcanic in origin, came up out of the ocean, and they've never collected. Anything that is there now had to have gotten there, either by flying or swimming or rafting, or brought in the last 450 years by humans to the islands, okay? So if we look at what is known of the islands, you can see the number of plants on Sao Tome and Principe. You can see that. Very few mammals are found on either of the islands. Birds, there are a few there. Amphibians, and you can also see the percent endemism. That is, those organisms that are known only from these islands. And there's relatively low percentage of endemism in the plants, about 30%, 18 to 30% in the mammals and birds. 100% of the amphibians, apparently it was really difficult to get there. The amphibians that got there a long time ago and then speciated on the islands. 100% of the amphibians are endemic to the islands. And here's what we knew about the fungi when we first started. Nothing. So the idea was to go there. I chose this in 2006 because my colleagues at the California Academy of Sciences had been working on these islands for many years. They had the project set up, they had the infrastructure, they had the connections, we had the government connections, we had the university connections, we had the connections with Belgium and the conservation people and with Portugal. So it made it easier to actually go there and to work. I didn't have to do anything with the connections. So I tagged along with the folks from Cal Academy of Sciences to be the first mycologist there in a long time. A couple just things to show you. This is an endemic begonia. That leaf is over 1 meter in diameter, if you know begonias. So it's absolutely fantastic. And they also have Sicilians there, not the Italian Sicilians, but they have Sicilians there, which are these legless amphibia. And so here's one here. It looks like a worm. It feels like a worm. It's bright yellow on top and lavender on the underside. But if you look at the end of it, it's actually got a mouth and an eye and a little nose. It's basically a legless amphibian. Really cool. You break apart the banana stumps that have fallen down, and these things will just kind of crawl out. Some really pretty cool endemic organisms. Okay, prior to 2006, when I went there, only 4 species of mushrooms were ever known from the islands. And these were reported in 1890 by Bresadola and Rominger. That was it. I went in 2006. I was the only mycologist on this expedition with the folks from Cal Academy, and I discovered 88 species of macrofungi that belonged to 52 different genera, and I estimated at that time that a high percentage of them might represent new species. So that was the good news. We found quite a few mushrooms and a number of belonging to a number of different genera. I thought this would be really cool. Well, the bad news was I had to pack up the specimens to get them home, put them in my suitcase, along with all the permits and everything else to allow them to come into the country. And the Air Portugal lost my suitcase for 5 days. So I came back into the country without my suitcase, without the specimens. Air Portugal sent them back. They entered into Newark. Newark opened the suitcase and said, what the hell are all this mushrooms doing in here? And they took them out and burned them and threw them all away. So a whole month in the fields, all that work for pretty much nothing because I had no specimens. It's kind of hard to get DNA out of a photograph, right? So yeah, that wasn't any good whatsoever. So what ended up happening was, as I said, the expedition was pretty much a waste other than they gave me a feel for what was there. So we had to pretty much do it again. And with at the contributions of a number of philanthropic individuals, one of the guys that ran the Dr. Robert Drews, as you can see, and I were going around giving talks about this and how it was a real problem. A couple of friends from the audience popped up and said, well, what's it going to take to go back? And so we said, well, we need some more fundings. And they said, how much? We gave them a figure. They pulled out the checkbook and wrote his personal checks to go back. So philanthropy got us back to the islands. And in 2008, I went back again. So this was our leader. That's Dr. Robert Drews on the right there. That's his field attire. He's a herpetologist. He's the expert in the reptiles and amphibian of Africa. He did most of his work in East Africa. He spent a lot of time on Principe as well. And he's also a birder, but he has no sense whatsoever. He's just standing in a mud puddle looking up at them. Just one of my favorite photos of him. So he was responsible for getting us here. He's been there about 20 times doing work, described a lot of new, almost all those amphibians that you saw there. Here was the team that we went in our little group. Anyway, a couple of botanists, a photographer who was taking pictures of the expedition and for educational purposes for Cal Academy, Bob Drews here, and there's a relatively young Dr. Brian Perry there right in the middle, who's supposed to be, if you're listening, Brian, you're supposed to be here right now. We had really terrible accommodations when we were in Principe. This is where we had to stay. Actually, it's owned by a billionaire from Belgium, and he allowed us to stay at his resort for nothing, basically. We chose one of these as a place where we stayed, and one was our laboratory, basically. While you're there doing the field in the tropics, you have to get around. And the best way to do it is to rent vehicles or borrow vehicles. When I did the work in Hawaii, I borrowed the vehicles from the Nature Conservancy. I wrecked one of the vehicles, drove it off the side of a mountain on Kauai. Yeah, but here we didn't wreck anything. Land cruisers and pickup trucks to drive over roads that, if you're lucky, kind of look like this through the jungle, but more likely you're like this. So everything is under construction there. When you're doing the sampling for the first time in the islands, as I said, we only knew about 4 species that were there. So we had to try and sample as many different habitats as possible. So we would sample a coastal lowland elevation like this. That is a baobab tree. Have you seen those? Or you know them from Madagascar, probably. We also collected mid-level, mainly secondary forests at the mid elevations, because the last 550 years of habitations there, people cut down a lot of the forests for various reasons. And now this is secondary growth that came in. This is Obo National Park. We did a lot of collecting there. But my favorite is to get up higher in elevation to the native primary forests, climbing up the sides of those mountains that I showed you in the first couple of photographs, and getting into habitat that looks a lot like that. Walking through streams, another beautiful baobab tree there. If you're lucky, there are some sites with established trails. There's one trail right there that work. This is Bob Drews right there. He was collected a few cobras while we were there, which was kind of fun. But most of it is walking through forests like this, where there really aren't any trails. You bring a guide, of course, and you have your GPS. So you hopefully won't get lost. So how do we do it? Well, basically, when you go to these areas that literally no one has gone and collected mushrooms before, at least for scientific reasons, maybe for the table a little bit, although they don't eat mushrooms, wild mushrooms in Sao Tome, we found out. But nonetheless, what we would do is we would go out in the morning. So you get up, have your breakfast, about 8 o'clock or so, you hit the road, drive an hour to where you're going to collect that day, about an hour. And then you'd go out and you'd collect for 4 to 5 hours. You do that 6 days a week. And I gave everybody 1 day off to rest. 6 days a week, 4 hours in the morning collecting. Eat your lunch in the field, drive back to the laboratory, which is the room you're staying in, wherever that is. And you spend the next 6 to 7 hours taking notes, photographing, preserving the specimens, etc. If you're lucky and you're fast at it and spend a lot of time doing this, which I have, I can take the notes on a specimen and photographs and get it all ready to preserve in about 20 to 30 minutes. It might take you a little longer than that. So if you're lucky, each person can maybe make a good 12 to 15 specimens per day. That's it. And that's a lot of work. So we're up at 7 in the morning, out in the field. At midnight, we would quit, and we do that 6 days in a row. You're spending thousands and thousands of dollars to get here and a lot of effort. So you make as much as you can of the time you have and the specimens that you have. You really need to preserve them really well. So here's our laboratory right here. You notice the mycologist aid in the middle up there as well. You need some of that to help you make it through the day. So you basically set up your apartment, your hotel room, resort room, whatever it is, into a make-up shift like that. And then you sit down and you start taking notes. So it's really important that you take notes on these specimens to make them valuable specimens so you can at least identify them later, because that's the whole purpose of documenting diversity, right? So we would draw every specimen, take notes on the cap and the gild and the side, all of that, have an accession book, color guide right here, color book. So we would denote what color it is based on the page, the column, and the row, and words that go along with that, like brown, brown, dark brown, grayish brown, etc., etc. We brought a lot of petri dishes with us that we would preserve the specimens in so that they don't get crushed and transporting back. So, and then we would take photographs. So notes in the laboratory, photographs in the lavatory. And here's Brian. The reason we chose the lavatory for a lot of the photographs is because it didn't have a window. You could close the door and make it pitch dark. And so when we're studying bioluminescent fungi, we would have a pure black, dark environment so we can have the light shots and the dark shots. So we would do that. Yes?
One seat.
Well, we have to go. We move them. And then all the specimens, after you spend all those hours of getting them ready and everything, we dry them. And I will do a product statement here. Nesco American Harvest Garden Master Dryer, best one in the world. And the reason I'll say that is because the electricity in Africa comes and goes. It can go anywhere from 110 to 240 from the same outlet. And it can be on or off anytime. So at some point, this sounded like a popcorn machine as electricity turned on and off, on and off, on and off, on and off, and it did not fry out the dryer. It actually stayed and has been working for years now. So I swear by these. We dry the specimens. You notice an accession label goes there. Petri dish to keep them when they're dried so they don't get crushed. Put them up in baggies, put some silica gel in the baggies, and then all ready for transport back. And then at the end of the day, of course, you might pull out one of your beautiful little cup fungi and pour some Valentines into it and have a little sip. Okay, that was the fun part. That's the fun part of documenting diversity in the tropics. Now the hard part starts, and that's actually trying to identify what the hell it is that you found. And this is where that really takes a lot of time. I want to spend a little bit of time talking to you about identifying specimens, what it actually requires and how to do it. First, you have to choose a species concept. I'll explain that in a moment. Then you have to understand nomenclature and taxonomy, the difference between them and the melding of the two in order to put a name on them. I'll explain that. We've already generated our macro descriptions in the field. You need to generate micromorphological data in the lab. You need to generate molecular data in the lab. You need to have access to all the available taxonomic literature on a particular genus that you're trying to identify, the species in that genus. You need to borrow and study type specimens. I'll explain those in a second as well, and other representative material of species you think might be what you have. You need to compare your data with the specimens of all species that have been described in that genus so that you can help identify it. Conduct phylogenetic analysis to tell you a little bit about who's related to whom, which will help narrow down your search for a name for your species. And then you have to make some taxonomic decisions. And I think the biggest take-home message tonight is going to be taxonomic decisions and what that actually means in the field of mycology. So, what is a species? There are a number of different ways to try and decide what makes a species a species. And there are three major ways to look at it. And these we call species concepts. They're morphological species, biological species, and evolutionary species. Morphological species are basically based on shared overall similarity in morphology or physiology, ecology, etc. Anything about the morphology and its placement in nature that you can see with your naked eye and document. And then all organisms that share all those characteristics, you could say, would belong to the same species. So it's based on overall morphology, shared morphology. Biological species are based on the ability to exchange genetic information. In other words, interfertility. If two organisms can mate and leave an F2 that is viable and then go on and leave more offspring, then they belong to the same species. And if they're intersterile, they cannot do that, then they belong to two different species. So that tells you that you have to be able to test that by being able to take two mushrooms. I mean, you can't just take two mushrooms and bang them together. That isn't how they mate. So we typically almost never have biological species information to address that type of a species. But it can be done with species, and I've done it with a number of species. And lastly, could be an evolutionary species, which is based on genetic relatedness. And this is typically derived from sharing sequence data. Individuals that share the most amounts of the same sequences of the same genes more than likely shared a very recent common ancestor and belong to the same species. So all of these species concepts, the only way you can choose a species concept is based on the amount of information you actually have or can obtain. Most mushroom names that are out there, most mushrooms are based on the morphological species concept. It hasn't been until the last 25 years that we've been able to generate molecular data and then use a combination of morphological and evolutionary species concepts to put names on our species. And now that's what we do. Nomenclature and taxonomy. These are different as well. So we're talking about identifying species. That means you have to put a name on it. Well, how do you do that? Well, there's two components of that. Nomenclature is how you assign a name to an organism, and it's governed by a whole set of rules that are in the international code for algae, fungi, and plants. We just call it the code or the ICN. It consists of 62 articles and lots of recommendations that accompany those articles. Okay, so there's a code out there that tells you how to name something. All right. The application is determined by nomenclatural types. So each species has a particular type specimen that name is attached to. And the taxonomic group in which it belongs to, whether that's a species or a genus or a family or order, whatever it is based on the priority of publication. So the oldest name at rank is the name that has priority over all other names that may be the same species that were determined later. Okay. And the names that you are choosing here, they have to be effectively and validly published. And there are, 11 different articles which tell you how to effectively and validly publish the species name. Okay. So the key here is the code provides rules on how you name something, but not what you name it. Okay. That's really important. Taxonomy is basically the science, as it says, of defining species and organizing them into hierarchical groups. All right. So what you have to do in taxonomy is first define what your species concept is. Do you have enough information to just do a morphological species? Do you have mating data? Do you have shared genetic data? Do you have molecular data for evolutionary relationship data or not? Then you have to define your characters. A character could be the cap color. And then the character state could be the color, brown, red, green, blue, whatever it may be. Okay, and that goes all the way through, different forms of them. You have to then place your organisms with similar features into the same taxonomic group. You have all of these characters, you have all these characters, they look all the same to me, you all belong to the same species. You do it that way. And then you have to base your taxonomy, put them into higher groups, into the genus and the family, etc. And then you base this taxonomy on evolutionary relationships, those phylogenetic relationships you have determined through molecular data, basically. Now, here's the key to this part of taxonomy. There is no governing organization that determines the correct taxonomy. That is done by a consensus of the mycological community. Okay, whether I think this thing is the same as that thing there is my taxonomic concept. You don't have to agree to me. You will see now in the literature that the boletes, we all love boletes, the boletes and what used to be all in the genus Boletus is now into about 35 different genera. So they've carved that up into a whole bunch of different genera. Now, most everything you call Boletus is not Boletus now. It's Butyriboletus or Pseudoboletus or Rubriboletus or on and on and on and on and on. Okay, that's because the molecular biology set up a huge phylogenetic tree and showed all of these separate little lineages, these clades, that someone decided, well, we can understand them better if we break them all up into separate genera as opposed to just call them all Boletus and belong to one group. You don't have to believe that. You can call them all Boletus if you want to. That name is still valid, etc. But the current consensus is that these all belong to separate genera, and we're going to call them by this name. So it's done by community, and that's important. Okay. Species names are based on type specimens. So as I said, by definition, a type is a single collection that was made and given to the author of that species at the time that they published the paper, and he based or she based their concept of that species on that single collection. There may be a lot of other collections they looked at as well, but a single specimen collection was designated as the type, and the name is tied to that specimen, to that type collection. This is really important, okay? And to belong to a species, a specimen must match the features of the type. So if I have something over here and I think that is the same thing as a name associated with this species, it has to have the same features as that type specimen. Is it clear? All right. Each genus has a type species that is the type of the genus. So if you're going to belong to this genus and you're a species, you got 15 species, where do I put them? They all have to have features similar to the type species of that genus in order to belong to that genus. So the type species of Amanita is Amanita muscaria. So you need to have some features of Amanita muscaria to belong to the genus Amanita. And in this case, there's only a few that overlap all species of Amanita, like the universal veil, white spores, smooth spores, et cetera, et cetera. Okay? Another key point here, the amount of variation allowed to delimit species, gender, and other taxonomic characters is determined by the taxonomist based on available data. That's important to keep in mind as well. And I'll just give you just a real quick, I'm going to go through this real quick to show you how you tie nomenclature and taxonomy together to arrive at a currently accepted name, a name that should be quote, correct. And we never use the word correct name. At least if I do, correct me. Okay, here's an example. So way back in 1860, a paper was published by Barclay and Curtis, and in it they described Agaricus californiensis. Okay, this was based on a specimen that was collected by Charles Wright somewhere between 1853 and 1856. And he was part of the North Pacific exploring expedition that had these boats that went up the Pacific, and they came down, they sampled some areas in California. They stopped at Mare Island, which is over in Solano County, if you know where that is. They stopped at Mare Island. Wright went out. He collected a little mushroom from Mare Island on coastal live oak trees. And he dried it, and he sent that specimen off to Berkeley, and Berkeley decided it was a new species, and he called it Agaricus californiensis. Here is a photograph of that type specimen. Okay, you notice that it is not great condition. So what Wright did when he was in the field was he cut some pages out of a book. He took the cap, he pressed it flat on the cap like that. Here's one specimen still attached to the leaf. He had it on the side. He put them together in this little packet here. He took some notes on it here. Right here, these are still stains from the mushroom. It was all in the packet there. And then he shipped it off to Berkeley. And this is what we call the prologue. The complete description of this new species basically says the pileus, or the cap, is short and conical and bell-shaped with a stipe that is gracile, so it's kind of thin, and all of it is red. The lamellae are free and white with a reddish margin. That's it. That's all the data. So if you've collected these little tiny mushrooms before, you could probably think of a lot of things that kind of look like that. That's the entire prologue. I borrowed the specimen from Kew and studied it, did all the microscopy on it and everything. So I had a pretty good concept of what Agaricus californiensis was. And the micromorphology says it belongs to the genus Mycena. So this is really a Mycena, okay? Actually, Barclay said so himself. He even wrote Mycena up there after A, which stands for Agaricus. So he hinted that it was Mycena. So back in 1887, Sicardo comes along and says, well, Barclay thought it was a Mycena. I'm just going to transfer it into Mycena. And he formally transferred it to Mycena as Mycena californiensis. So now we have two names for this, Agaricus californiensis and Mycena californiensis. Which is the correct name? They're both correct. All right, and they're both valid, they're both effectively punctual. Which would you want to use? You'd want to use the one with the characters that says more likely what it is, which is Mycena. Okay, so those two names, Mycena californiensis and Agaricus californiensis, are homotypic synonyms. They're based on the same type specimen. Okay, following me so far? All right. Not too long ago, actually, Brian Perry, this was his second published paper when he was working with me at State. We were looking into this beautiful species here that all of us at the time knew as Mycena elegantula. You go back to the field guides from California, David Aurora's book, and all of the other ones that predated the 2000s, and you'll find Mycena elegantula in it. Well, Mycena elegantula was actually collected in the late 1800s. It was published by Peck in 1895. It was collected on California live oak leaves in Pasadena by Professor A.J. McClatchy. He sent it off to Peck at the New York State Museum in Albany, New York. Peck described it as a new species, Mycena elegantula. I borrowed the type. I looked at the type. I compared it to Agaricus californiensis. They're the same species. We sequenced the material from Southern California and from Pasadena and from the area around here near Solano County. They're all the same thing. So this name, Mycena elegantula and Mycena californiensis, represent the same species. Based on priority, the oldest name in rank is californiensis, dates from 1860. Elegantula dates from 1895. So the best available name for it is californiensis. It's a Mycena, so it is now known as Mycena californiensis. And that is taxonomy, saying that Mycena elegantula and Agaricus californiensis are the same species is a taxonomic decision, not a nomenclatural decision. Which name is the appropriate one to use is a nomenclatural decision. That makes sense? Take a little longer. But anyway, that takes a lot of work to figure that all out. Okay. So when you're going out and collecting, and you can all help do this as well, particularly for this fundus project, is to collect your specimens. If you think it is really unusual, it isn't just, we don't need more specimens of anemone or muscaria. We've got enough of those, right? But if you think it's an unusual species, you can take it and you can start taking notes on it. Take notes on all of its macromorphology, all parts of the mushroom, the size, shape, color, surface textures, etc. Record the taste, the odor, the substrate, the habitat that it's in. If you can, make a spore print of it so we can see what the spore color is in deposit. Note any color changes when you bruise it or add specific chemicals like strong bases, things like that to it, and take these morphological notes. And then you can take these and you can upload those to iNaturalist, upload those to Mushroom Observer, etc. Dry the specimens and then make the specimens available to somebody who has a herbarium or a fungarium, either me at San Francisco State or Brian at CSU East Bay. We both have large mushroom herbaria, and that'll really, really help advance our science. And that's really important. That's what Brian was going to talk to you about here. Shameless self-promotion, buy my book. And if you go to Hawaii, buy my book. And then once you have all those macromorphological features, that's only one subset of what you need to actually put names on these species. You need to do the micromorphology. So if you enjoy this stuff, which I really do, invest in it in a nice little microscope and look at the gills so you can see what the spores are like. Note the spore morphology. There's just a few samples of some different mushroom spore morphologies up there. And note their ornamentation, their color, their shape, size, all that kind of stuff. Look through the cap surface, the gill surface, the stem surface, etc. for these cells that we call cystidia. They're any cell that's morphologically and functionally different from the cells that surround it. And you can note their size, their shape. Are they ornamented? Do they have some funny little contents that are very oily here? You can even hypothesize as to what their function is in the mushroom. So take those characters down. Every tissue has to be examined, particularly the cap surface and the stipe surface. So here, is it formed of kind of round or club-shaped cells in many layers? Is it formed of cells laying flat with lots of incrustations? Is there a pileus surface here and then a universal veil on top of it? Then you put it in Melzer's and it turns red. That's a dextroid reaction to this tissue, but not that tissue. Or the stem surface, does it have hair sticking out? Are they thick-walled or thin-walled? Are they a little spinny all over them? Et cetera, et cetera. So these lots and lots of microscopic characters that you can use. And then pull out your PCR machine and sequence them in your kitchen. Or give them to somebody who does that, and there are facilities in the Bay Area to do that for you now. Get the molecular data. So for fungi, the DNA fingerprint or the fungal barcode is the internal transcribed spacer region, and the 5.8 is a gene region in between those two. So you pretty much need to sequence the ITS region to even publish a new species anymore. And the ITS is a very variable region, and so we can use it to help delimit species. It's very good for that in some groups. If you're thinking more about the genus level or a higher order level, you need more conservative genes. So the large subunit or the small subunit is very good at the genus rank and above. And then sometimes the ITS won't help you very much. You need something that's variable, but also a little bit more conservative. So you can use the RNA polymerase 2 beta subunits 1 and 2, or the translation elongation factor 1 alpha, those gene regions to help you generate more molecular data to compare between species. And that's just to show you what it looks like when you get the raw data. And then you align them and you run your phylogenetic trees, and that will help. It only helps. It doesn't get you everywhere. You also need to know the literature. You need to accumulate all the literature that you can on the particular genus that you're working on. If you're working on Mycenas, here are a few books that you need to have. Unfortunately, not all of these have been scanned. They're not all available for AI. Don't trust any AI identification yet. It's going to be a while before you get to that. There is more literature available now online than there ever was before, because people are scanning these and are uploading them. And some cases you have to pay for them. Some cases it's free. I'm a bibliophile, so I just buy every mushroom book I can get my hands on. And now I don't know what to do with them. My lab at San Francisco State and our library's got about 20,000 books in it. It's absolutely incredible and no place to put it. And they haven't rehired a position to fill my position. So I was going to give it to the next mycologist, but they haven't deemed that they need one yet. All right. With that background in mind, now we're actually set to look at what it is that we used all those data to allow us to do, and that was document the diversity of an island that no mycologist had really ever gone to and studied before. And this is the most fun part about collecting mushrooms, is going to these underexplored areas where you may be the first mycologist that was ever there to collect. So let's just look, and I can go through these relatively quickly. I don't want to bore you. Here's some Mycenas. Both Brian and I love Mycena. Brian did his master's under me on Mycena, and we did the postdoc together on Mycenas as well. So we love the genus Mycena. We found 19 species of Mycena in the expedition. So if you recall now, this is just a 2008 expedition. We're there a little over 3 weeks. 2006, we lost all the specimens, but pretty much all these data are based on the 2008. 3 weeks in the field, 18 days, 12 hours a day working or so on the stuff. So 9 new species out of those 19. The rest of them are all new distributions. We named this one Mycena as a new species, Mycena solus, because it looks like the sun. There's those sun rays shining off the top. And I'll just show you some of the micromorphological features so you can get a feel for how different these things can look under the microscope. There's a couple more new species, Brunio violacea and Phaeonox. Just a bunch of Mycenas. Mycena allies, things related to Mycena, genus Hydropus and Phylloboletus. You notice the Phylloboletus, they have pores instead of they have gills, but then they have tubes inside those, which are just little cross gills, basically. And then you have to, in order to put names on them, you have to have all those characters. You got to look at all the literature. You run a phylogenetic tree, and there it is. This was a project that my graduate student, Lexi Cooper, did as her master's degree. And then some other beautiful little Mycena allies. This is Favolacea thwaitii. Again, this is more poroid, but it is a poroid gilled mushroom. And here's another poroid gilled mushroom, Mycena perella griseopora. And it has very, very tiny little pores, little tiny tubes underneath, but it's a Mycena. Gymnopoid is basically anything that looks like the genus Gymnopus. We found 31 species here in 14 different genera in that group. 10 of those are new species, and all the rest of them were new reports for the island. Here's a couple here. I named this one Rodhollii and this one Bilbosii, because I told you that we had some philanthropists that helped pay for our trip there. Rod Hall was one and Bill Bose was the other. So if you want your name to me. And some other ones. This looked like the iris of an eye to me with brown eyes coming out. So we called it Ocellus. And just a few more here. The genus Trogia is strictly a tropical genus, pretty much an old world tropical genus. We don't have any Trogias in the new world that I can think of. And the type of it is related to this one right here, Trogia infundibulariformis. Beautiful little things here. This one smells like garlic, Lysotinus ignobilis. And this one is a huge palmivorous. You can imagine what it eats. Yeah. On the right-hand picture here? Yeah, at A, those are the spores. Yeah, in all cases in the drawing, A are the spores, B here are the basidia, C are the cells on the edge of the gills, those are called chylocystidia. D is what the cells on the cap surface look like, all together in the cap surface. And E here are the caulocystidia, the cells on the side of the stem. And it pretty much goes in that order in most of the drawings. This one here was known for years as Marasmius palmivorus. It is a devastating pathogen of palm trees, and it's been known around the world as a devastating pathogen. Quite a few papers were on it, but it never did belong to Marasmius. And just a couple of years ago, a couple other mycologists put it in its own genus with one other species and called it Paramarasmius. So that's a new generic name. And this beautiful little thing, we published it as Pleurocolibia imbricata, And 3 years ago, based on molecular biology, some scientists in Europe said the entire genus Pleurocolubia really belonged in the genus Callistosporium. So they transferred this from Pleurocolubia to Callistosporium. So again, these would be homotypic synonyms, but the currently accepted name would be Callistosporium imbricatum. I hate using that name just because it doesn't look like anything else in Callistosporium at all. It's like apples and oranges, but that's where it belongs based on genes. And then some just glorious specimens. This is a pan-tropical species throughout the old world, Cyptotrema aspirata. And then, of course, there's just to show you the phylogenetic tree. When you do this, you can even kind of address levels of endemism. So this is a pretty thorough study of the gymnopoids, and we know them pretty well from other parts of the world. And about 32% of the ones that came from the islands are known so far only from those islands. But I have to have the caveat that nobody's really collected in the DR Congo or in Cameroon or some of the other islands that are the part of the continent that's only 250 kilometers away. So it's possible that some of these also occur in continental Africa. Genus Marasmius, we found 22 species, 5 new species in there. And here's just kind of what they look like. Some beauties in here. Just whip through some of these. You can say they all have these beautiful cells that we call broom cells because they look like a broom. So one of my favorite ones, I'd never seen a species of Marasmius that had bright orange gills before. Here's the cap looking down on it. Here's the underside of the gills. So we named it Marasmius laranja, which in Portuguese means orange. It's just a beautiful specimen and large phylogenetic tree here. This work was the master's thesis of my student, Chris Grace. And then one of the most exciting things for me was this genus Campanella. It's a tropical genus. Most of you probably don't know it, but it's been a big mess for a long time. And the reason it's been a mess is because nobody knew what the type specimen looked like. The type species look like. And so if you want to throw something in the Campanella, it has to be like the type species of the genus, which is Campanella buettneri. But Campanella buettneri was collected from a single specimen from Togo. The Togo is about 400 kilometers away from Sao Tome. There's no material that anyone's collected in that whole region of equatorial West Africa. So we had no specimens from Togo. But we had this thing that we collected many times from Sao Tome, and it matched the protologue, it matched the description of the type species of the genus Campanella. So basically, we redescribed buettneri based on this material from Sao Tome, which is pretty close to Togo. Well, it's not there. That I personally think is the same thing as buettneri. So we redescribed this. We designated this as what we call an epitype specimen. We've sequenced it. So now we published a paper on it. So now, if you believe me and you want to follow me, the genus Campanella is based on this species. And if you want to belong to Campanella, you have to have features that are shared with this species to be Campanella. And you throw it into a phylogenetic tree, and there's a whole bunch of other things that people call Campanella that surround and that are right there. So we rediscovered and redescribed the type species of the Campanella. We did the same thing with the genus Tetrapyrgos, which is described from Madagascar. And so we got material that my student collected in Madagascar, and we did that as well. So that really helps us nail down the taxonomy. You all know hygrophoroids. We only found 5 species. A couple of them, 3 of them were new, and they look like that. And Pluteus, most of you know Pluteus as well. We had fun with this genus. It's not an easy genus. Elsa Valinga is a master on this genus. But we did the work on it, did the micromorphology, sequenced them, etc. And we found 6 species. 2 of them are new species. This is one of the new species here, Pluteus hirtellus, this beautiful hairy brown cap with the cells on the cap surface with brown pigment on the inside. That's what the cross-hatching suggests. And then others with really fantastic cystidia. Look at these cystidia. I mean, these are just a big thick wall with horns on them, etc. Big lumps on them. Keep your eye on that cystidium right there, because Brian thought that it looked like Sid. So we started calling them Sidstidia instead of cystidia. And phylogenetic tree shows us where they belong in the greater scheme of things and who they're most closely related to, and also helps assure that the one we collected or the two we collected from South America fall right into the same thing with others that other people collected from other parts of Africa in that other areas back east. Dark spores, you can see some here. This is the one that you saw in the drawings that was drawing. It's actually got Physospora with the beautiful spores with a roughened exosporium here in a little smooth area called plage. And Crepidotus, this one here reminded me, you ever see those newsboy caps? You know, the little flat caps that I like to wear? This reminded me of a flat cap. And Kangol makes the flat caps as the company. So I called it Kangoliformis. I sent the paper off to the makers of Kangol, hoping they'd send me a free cap since I just named a mushroom after them. Never heard back, as expected. Some coprinoids here. You all know the genus Coprinus, the Coprinopsis afronivia. That's a new species there. And this beautiful one that was actually described from Papua New Guinea, but we actually found it up in South Tome. It's really a nice species. And some more Satharellas here, Diconicas, little black spored. Here is the tropical version of your sulfur tuft around here. So those of you who know Hypholoma fasciculare, very, toxic, very bitter species. It's much larger. If you can see the size of it, that's only 10 millimeters, the entire cap. So it's really a little tiny thing. And it just grows all over the side of the logs. This is Hypholoma subiridi. And it's probably a new species, actually. The molecular data shows it up here, which is pretty much sister to this clade fasciculare, and the real subiridi, which is a New World thing. So it's probably something new. So we just called it AFF subiridi. A few lepiotoids, which we haven't worked up. Not my forte. Beautiful little lepiotoids, though. Leucocoprinus here. Another Leucocoprinus. One Amanita, one Tylopolis, and this one. Now look at that. You people have collected mushrooms, right? You know coral fungi. If you were to look at that, what genus would come to mind immediately? Just somebody. Ramaria, right? You'd think Ramaria. Ramaria is an ectotrophic, so it's mycorrhizal with various trees. I looked at this and said, oh, wow, they got Ramaria here. That must mean there's some ectotrophic tree around here, something that can host this mycorrhizal symbiont with it. And I looked around and didn't see any mycorrhizal trees, which was interesting. So I thought, okay, it's a Ramaria. So I took my notes on it, brought it back. We worked it up, and Brian sequenced it. And I was willing to see, I was looking at the spores of the thing and I'm going, those don't look like Ramaria spores to me. They look much different than Ramaria spores. I thought they're too small for that as well. A lot of Ramarias have spines like this, but they were way too small. This must be the smallest spored Ramaria species ever. Did the sequences of it, and he said, Dennis, this thing isn't even near, it's not even the same order as Ramaria. This thing belongs to the crust fungi. You know crust fungi, those little thin slimes that are on the logs? This thing is a coralloid crust fungus. Who would have guessed? So we did the phylogeny on it, and here's again nomenclature and taxonomy. You see it up at the top there, Ctenopogon. I named it as a new species, Havencampii. Ctenopogon havencampii. It falls with another coralloid Ctenopogon species, but it's sister to this genus Trachyspora. It's embedded in Trachyspora. So at the time we thought, well, we should probably just call it a crust fungus, even though it's coral shaped. We should call it Trachyspora. But at the time when we published this in 2015, the type species of the genus Ctenopogon was unknown. It was never sequenced or recollected. It's called Palescens. And the type species of the genus Trachyspora, which is Onusta, I believe, it was not in any of the trees, and it was not known either. So if you don't know what the type of this genus is, you don't know the type of the other genus, and this thing fits in the genus with other things that are called Ctenopogon, let's just call it a Ctenopogon until more data are available. So we did that, and then back in 2021, I think it was, some Europeans were working on this crust fungus group, Trachyspora. They sequenced a bunch more Trachysporas. They actually found material of the type. They threw it in for Trachyspora. As it turns out, some of the other coralloid ones are embedded in different areas within Trachyspora. So it was obvious that Ctenopogon belonged in Trachyspora, and Ctenopogon as a genus was a synonym of Trachyspora. So basically, as it turns out, these crust fungi just decided to lift up and turn into little corallite fungi independently a number of different times over the evolution of this lineage. Really interesting stuff. So this is now called Trachyspora havencampion. Again, homotypic synonyms. All right, so to conclude then, in this 2008 expedition, we discovered 188 species of macrofungi that belong to 80 different genera. I went back to my notes from 2006, and from those I could determine what was a distinct species, how many different distinct species we have. I couldn't put names on them because I didn't have any material. So I couldn't say what the species was, but I could determine kind of what was there. In 2008, we recollected 41 of the 52 genera. So we were able to recollect 79% of the genera I saw in 2006. And we were able to only recollect 58% of the species I found in 2006. So this snapshot of 3 weeks of intensive collecting only got a little snapshot of what's really there. And even the snapshot of 2006, which I don't even know what those species are, only gave a small percentage. Add them up together, so far, diversity is somewhere around 225 species and at least 91 different genera of fleshy fungi that are there. So in order to get a real handle on what's in there, you need to be there. You need to be there all year long. You need to collect often. You need to do it more than 1 year, on and on and on. So you need to be a local to do it. And we've been training some locals from the area. The biggest surprise, about 22 to 53% of what we found we can call putative endemics, known only from these islands. 4 of the species we found are putatively ectomycorrhizal fungi. We found them only on Principe, that one Amanita, the one Talopolis, we found one Scleroderma, and one Russula. But none of the trees are known to be ectotrophic. And we talked to lots of botanists. There's a good flora. There's none of the trees there are known to be ectotrophic. So what the heck is going on with those? I don't believe they're saprotrophic species. I think they're ectomycorrhizal, but they are forming some relationship with some other tree that is probably native to Principe that we have never known to actually host mycorrhizal fungi. Another interesting fact was that many of the species that we found are very closely related to taxa from the Caribbean region. And there are several reasons that are possible for that. Depending on how old these lineages are, if they're more than about 80 million years old, in some of these lineages, which is kind of hard to say since the islands are only 31 million years old. It could have been because Africa split apart and South America and the Caribbean region used to be part of Africa a long time ago, and the ancestors could have moved and then they evolved from those ancestors. Or probably more than likely, where did the slave trade from Sao Tome go? And where did it go back and forth to? Haiti, Jamaica, the Caribbean. So a lot of these may have been relatively recent introductions, kind of mitigated by the slave trade from the Portuguese for the last 500 years. We don't know, but that's an interesting surprise. And then just lastly, I just put this up because I always like to say this. All of this type of work, there's almost no funding for it anymore. There is some administration in the White House that doesn't seem to approve of environmental studies or understanding diversity or health science for that matter. So all the funding of the NSF has pretty much gone away for diversity studies, which is not good. We have to get that back, folks. Vote. The climate is changing. That influences what can grow there. We see that in the Sierra Nevada from the 30 years that I taught the spring fungi of the Sierra Nevada. Brian Perry took that over in the last 5 or 6 years or 7 years. He's been doing that. We're seeing less diversity, less abundance. Interesting, this is drying out in these areas as the climate changes. Habitats, as we know, are disappearing because of deforestation, development, and now pretty much wars everywhere. Many governments are restricting you from removing any biodiversity from the area. Most of the work that I do in Brazil now, I pretty much have to do it in Brazil. They don't like us to remove any of their specimens out of their country, particularly if they're living, so no mating studies or anything else like that. The environment influences mushroom morphology. We know that. Mushrooms are short-lived. You have to be in the right spot at the right time to find the things. The names in our taxonomy of everything is pretty much based on the mushroom. Now, there is a move afoot, not one that I'm a big supporter of, but there is a move afoot to use ITS sequences as type specimens. In other words, you won't need a specimen. You don't need any material. You can sequence DNA out of the soil, find an ITS sequence that's different from everything else, and call it a species. Makes no sense to me whatsoever, other than documenting all the different operational taxonomic units that are out there in nature. But as it stands right now, mushrooms, you have to have the mushroom in order to know how much diversity you have and to put names on the thing. And those mushrooms are, they're hard to find. You have to be there at the right time. They change by the environment. It requires you to have access to obscure literature and access to historical specimens, so borrowing from various herbaria. My herbarium in San Francisco State does not allow me to mail specimens out unless I pay for it, because they have no funding to ship stuff out anymore. So I just shipped something to Brazil. It cost me $97 to ship it to Brazil out of my retired man recovering academic pocketbook. Species are based on type specimens. Many of the types no longer exist, or they're in a bad condition as you saw, and you can't sequence that bad specimen. Many of the specimens don't cooperate in the lab. You try and get a certain sequence out of it, they just say, eh. They thumb their little hyphae at you. When you do have that, and the molecular sequences databases, as you know, by definition, are incomplete and will be until we get everything sequenced and thrown in, and then you have access to it to see whether your sequence matches something that's already known. And lastly, it just takes a lot of time and a lot of hard work. We did the field studies in 2006 and 2008. We published the data in 2015 through 2020. So it took 8 years to 10 years to get this stuff out and studied and actual names on it. And we're still not done with that one project. So hopefully that gives you a feel, a little photo credits there. There's Brian in an awkward position, photographing Palmivorous. And I will thank you and leave you with this incredible fungus here. That is actually an aspergillus, if you know aspergillus, which is a mold. That is aspergillus, and it has an interesting story behind it as well. Thank you. If you have any questions, I'm still here.
Phallus drusei?
Yes, it was. I didn't put that one in. He's talking about a species there. We found a stinkhorn that belongs to the genus Phallus. Now, Bob Drews, the guy that ran the expedition, he's been wanting me to name a fungus after him for many years. And he was with me when we collected it. So I told him, beware. So as it turns out, this Phallus is the smallest species known. It's less than an inch long. It's the diameter of a pencil. It's the only one that curls down at maturity. So it's flaccid at maturity. And I named it Phallus drusei. I named it after him. It hit the airwaves, was pretty good. My friend Peter Sagal picked it up for Wait, Wait, Don't Tell and put it on the Bluff the Listener segment if you ever listened to that show. So it was great. It's immortal now. So yeah, we have we have fun with these here. I named one after SpongeBob SquarePants. I named one after my wife, and on and on and on. So yeah, Chris.
Early on, you mentioned that since those two islands occurred organically, some amphibians and maybe even mammals got their own rack. So those are like naturally occurring, like chunk of soil and
yes,
a bunch of logs.
Yeah, they documented this, particularly out of the Congo Basin.
So the Congo
River, it dumps out into the Atlantic Ocean, and then big wads of fallen trees and mud and other vegetation can just through tides can just raft all the way there. There's a lot of documentation on the same thing to the Hawaiian Islands. Now the Hawaiian Islands are 4,000 kilometers away, and there are very, very old cedar, Western cedar canoes that were carved by native Hawaiians in the Hawaiian Islands from logs, cedar logs that floated from probably Washington or British Columbia all the way to the Hawaiian Islands. So rafting is a thing. With a lot of fungi, probably there's a lot of vectoring on mud and birds' feet. There's a lot of migratory birds that do that. Probably the limited amount of airflow, jet stream flow of spores documenting. It's easier to understand how you can get an invasion or the migration of a species from a distant area if a whole packet of spores moves. Now, some fungi have been able to do this by designing their spores to cling together and move as a packet. But otherwise, if single spores move, it takes two to tango, and they have to be made incompatible. So you can imagine blowing, say, to Hawaii, 4,000 kilometers from California, right? We have some species there whose ancestors are from the Western, the US. Spores would float there by themselves. They would have to land. The spore would have to germinate into the hyphae, the monokaryote hyphae, and then it would have to relatively soon find a mate that was mating compatible with it. So what are your odds there? Either 50% or 25% based on your mating system that you're undergoing, right? So the chances of that are really slim. However, if they came in the mud and they clung to a bird's feet and the bird migrated there, you could establish a population pretty quickly. Yeah. All right, thanks for being so patient.